Oh, my name is Lew Lim. I am the founder of vLight. Now you know about red light therapy. Photobiomodulation is probably the right way to describe it. And we focus mainly on what it does to the brain. So this is really mainly about brain stimulation. It looks very complicated. It's colorful, it looks complicated, but actually, in a nutshell, what this is saying that is the red light and near-infrared light can penetrate to your skin. That's why red light therapy works. It gets into the cells in the mitochondria. It activates the mitochondria in a nutshell to activate the release of cellular energy, release nitric oxide back into the body or the brain and improve circulation. But the important thing actually is, in my opinion, what it does in terms of gene transcription. The release, gene transcription is modifying the proteins and genes. It allows physiological effects. And it does do some other things like reduction of inflammation. And also, in my territory, what's important is actually the release of growth factors, which allows the brain to regrow its volume, after attenuation, like the neurodegenerative diseases, stuff like that. How do we know it works? There's always this question: light penetrate your skin, your brain, and I can spend like an hour talking about the mitochondria and the mechanisms, but the crux is really to see that the brain responds. So we have an ongoing study in Toronto at Baycrest Hospital where we actually see what the real-time effect of the brain is to something on the head, right? And this is what it shows. This study is ongoing. Now the device could be like this. This is the consumer version you put on your head. Just slip it on, and I don't want to waste the seconds putting on, take it out. This is basically it. But the end result is that if you point, in this case, a laser, it has to be MR, the scanner compatible. Just one here and one in the nose, and you get a global response from the brain in terms of blood flow. Now, if you look at the literature for other forms of brain stimulation, even other types like magnetic or electrical, you won't find the kind of response that you see like this in terms of blood flow, cerebral blood flow. They call also the other type, call it BOLD, which is blood oxygen level dependency. So this is ongoing. These are preliminary data. We are waiting to finish this study on 42 subjects around summer this year. So what we see in MRI findings is the brain response to PBM is really quick. The intranasal has disproportionately large effect. Think about it. There's no barrier between the nose and the brain, really. No scarp, no melanin. And when we combine with that and our brain, it's pretty comprehensive. Now, the other thing that's important, and sometimes you read in the literature a few years back, wavelength maybe doesn't matter so much. Pulse frequency, the number of times it pulses per second, doesn't matter that much. Power density, maybe not. But what we are discovering is all these things matters. It will be published. Hopefully, within this year, you see a number of publications. Some of you who do neurofeedback are familiar with EEG. Here's the other way to look at it too. Now, how does the brain respond when, in terms of electrical signaling, electrophysiological signaling, when you induce a certain frequency in the brain? I'll cover a bit later. Now, we want to say upregulate endogenous gamma in the brain. Why? We'll cover that a bit later. So we induce gamma, and the first row really is nothing, is the baseline. The second row on the left, you see, is 5 minutes before treatment. The third row on the left is what happens in real time. And then the fourth row on the left is 5 minutes after you stop treatment. Then 25 minutes and half an hour later, at the bottom left, you see that the brain reactivates even half an hour later. Now, the right column are really the net effect, that treatment minus the baseline. You see 5 minutes before, okay, this guy is pretty low gamma before the actual treatment. And during treatment, you see on the right column, the right side, the gamma shows up. So we induce gamma in the brain. We induce gamma frequency to the brain. The gamma actually builds up even after you stop. Okay, 5 minutes later, it went back down, but half an hour later, it goes back up. There is a longer term effect after we induce a certain frequency in the brain. Very interesting for those who are doing neurofeedback because you're trying to influence the brain waveforms. What about cognition in normal, healthy people? There are actually quite a number of papers. This is a review, a collection of papers, but just in summary, it shows that even healthy college students actually respond in terms of cognition, quicker response to challenges and stuff like that. So healthy people, there is something, but the studies are small. We are doing more in our lab. Oh, okay. I don't know how many of you follow UFC. Ilia Topuria won the World Featherweight Championship about a couple of months ago. He uses this, when you do contact sports like that, you get a lot of repetitive traumatic brain injury, right? But he is an example where the mind is very important. I'm going to be covering this in my short talk tomorrow. I'll try to break down the case in using it. I'll try to break down why he was successful in using this photobiomodulation in his brain to achieve what he achieved. The mind is really important. In sports, it is being used by the US Olympic team members. In Canada, this elite sports in skiing and a bunch of other people in hockey are using this technology right now. So we've been used by word of mouth. We don't do a lot of advertising and promotion, but he's going to support us. He's going to be an advocate. So we'll see. Now we do, apart from healthy brains, we do quite a lot of research into medical conditions. This is a list of the clinical studies that we do. Traumatic brain injury, dementia, brain fog and long COVID. We're wrapping up that pilot study. We're collecting data. Addiction, craving, depression, bipolar, autism, pain. But I just highlight yellow because I'm going to talk the first three. There's no time to talk about everything. I don't know how many of you have a vLight device. You got an earlier one. I apologize because we keep innovating and developing. And the latest version was the one I just showed you, the Neuro 4. But the point is we try to make it more comfortable, better contact. We have an additional module at the back of the head to stimulate the cerebellum. But we will continue to innovate and try to improve. Yeah, we have other things in mind, but this is actually the state of the art for us right now for a consumer home use device. I'll talk about the professional device a bit later. This is now the actually say, white one doesn't look cool, so we now have it in black. But the options available is set at 10 hertz alpha, 40 hertz, and then a combination of alpha and gamma, 10 and 40 hertz. Ilia Topuria actually started using intranasal only. He was popping all over Instagram, showing this, like the picture over there. And this tiny device here, You can have options of putting 4 different applicators. You can have 2 advanced. And there is intelligence in here to sense whether you're putting the near-infrared light, the same one that you put in the head, but this is just intranasal from the nose to the brain. And you can actually switch between alpha and gamma, but keeping your finger on this button. So there's a lot of options. It's portable. It's very affordable. Now I published this paper about a month ago for traumatic brain injury. You can take the QR code or you drop out booth, and this actually is for me to tell people that traumatic brain injury is really complicated. That's why there's no standard treatment for traumatic brain injury like a drug or standard protocol. And I believe if you read this paper, the many facets of pathophysiology is like what's creating the physiological changes in traumatic brain injury. Photobiomodulation is just one delivery, but it actually intervenes with many facets of what's causing traumatic brain injury symptoms. So that's in a nutshell. If you love, physiology and what's going on in the mechanisms, just go there. I think it's really getting circulated quite a lot. This is from the paper. It shows that there's multifaceted, from mitochondrial dysfunction, or reactive oxygen species building up, axonal degeneration, growth inhibitors. There's autophagy, autophagy is your normal cell dying and clearing up by the lysosome, the organelles in the cells to release enzyme to clear this debris in the brain. Apoptotic cell death is programmed cell death, is under control. Inflammation, excitotoxicity. Axonal injury is really the primary pathophysiology in the brain, because the axons are really disconnected from trauma. But just go there. It takes a long time to go to detail. Now I want to talk about gamma. What happens, I touched on it earlier, to the brain. This is a study on 49 former athletes who's got repetitive traumatic brain injury. Just let's look at some of the symptoms in red, like the outcome for depression, PTSD, rehabilitation, sleep, erection time, and so on. They all actually improve significantly. And the effect sizes are very significant too. So and when more and more materials come up, I think you understand that photobiomodulation could be an answer to traumatic brain injury when there is actually none at the moment. The protocol is rest. The modified protocol today in sports is, okay, you can start doing gradual exercises, but don't do anything to the brain. So hopefully this is, by the way, it's actually approved by the VA. I'll just touch on that later. Now, repetitive traumatic brain injury can develop into, in the long term, CTE, which is degenerative. You hear about retired athletes committing suicide, right? Losing behavior, is like dementia. A lot of work is done in Boston. They keep this largest brain archive, bank of brains in the country from ex-athletes. So Margaret Naser, the professor of neurology, research in neurology, over there did a study on 4 retired athletes using photobiomodulation. She was very thorough. She looked at least 8 symptoms. And I don't want to spend too much time, but just to show you the trend, just observe the trend. The top left talks about PTSD. So a reduction in the symptom is good. So it's trending down is good. Depression trending down is good. Pain and effect interference with family and community activities. That's good. So in the study, they did 1 week of photobiomodulation, and then they improved, study over, go home. But the alarming thing is when you go home, they actually regressed. They got back the symptoms again. And they got worse, for 2 months, they got worse and worse. But then Dr. Nazer said, we really got to have a home use device. So the vLight device was used at home. And you know what? You can see from after the second month, the improvement resumed again, which means that for a degenerative disease where I think your genome is being influenced, you have to do something like this regularly. But the good thing is, it's home use. It's a one-time investment in the device and no side effects. Today, one of those subject number 1, Larry Carr, is a strong advocate for this device because he said it saved my life. So you can go Google Larry Carr, C-A-R-R. He'll have, I think, video testimonials and stuff like that. He's trying to make an effort to get into the NFL, try to go on something like this. Then what about alpha? Why do we need alpha? I want to go back to traumatic brain injury in this case. Here we did a study with UCSF with Professor Linda Chow, who is a professor of psychiatry and neurology, and radiology. So she had access to all these brain scanners. Now, this is an active hockey player at the time, but he had repetitive brain injury. He got, kept being demoted. But he did this treatment. I'll just put it in a nutshell. The alpha worked better for him at 10 hertz. And after 8 weeks, not only he improved, but you look at the imaging, he actually had his brain volume, the gray matter, in particularly the hippocampus and the thalamus. Hippocampus is dealing with the memory. Regrowth was unheard of. It was a landmark discovery, actually. We see more and more of this. In fact, Larry Carr, his brain is, he did brain scan, we're analyzing the data right now. And actually, this study at Boston University is complete. They're analyzing the data right now on a control study. So VA has approved this technology to treat traumatic brain injury. They had over 100 subjects that showed significance statistically to have improved. So I think you're going to probably hear about photobiomodulation technology, probably vLight with the VA and so on. It is not that easy to get VA approved for something like this. What about dementia? This is actually coming out of a paper that I submitted and is under peer review right now. This is just the structure of the paper focusing on, here again, the pathophysiology of Alzheimer's disease. I'd say TBI, traumatic brain injury, is complicated. This is at least as complicated. But the medical industry is kind of try to simplify it today and say, it's beta amyloid. To the drug, you use a biomarker of beta amyloid and you improve. But actually, as you probably know, if you're informed, that you walk around with beta amyloid in your brain doesn't mean you have Alzheimer's disease. Completely healthy people have that. So this is a really complicated schematic chart. Is a topology of what's going on in Alzheimer's disease. I had to do quite a lot of literature research to come up with this. You won't find actually a more detailed topology of what's causing Alzheimer's disease anywhere in the literature. I've looked at the genesis. Okay, what's causing it, from environmental factors to your genes and all these other things like your blood-brain barrier, your gut-brain thing, and it's all up there. If you I think this is recorded, right? So you can see this. Then the biomarkers, the second row is not just amyloid beta. There's just one of the markers. There's energy deficit, there's oxidative stress, there's the calcium overload, there's a big thing, inflammation. Then you go to the precursors, meaning you are losing your cognitive ability because of the loss of neurons and synaptic connectivity, and the loss of volume in the brain, like I mentioned before. Then you get all these symptoms. But what makes things easier for us to try to treat it is it gets presented in brain waveforms and the brain networks. So it gives us a chance to try and treat it. This paper is under peer review. We'll show that later. Hopefully it comes out later. And I'll have a second part that's going to be submitted on how we can treat the brain. But there are not a lot of clinical studies being done for using photobiomodulation in Alzheimer's disease. I have 7 papers here on clinical studies. That's all there is. Actually, there are a lot of animal studies, culture cells, and what's happened to the structures and everything. We do that ourselves. But this is just 7. But the common factor is these are ranging from anywhere between one subject, over the head intranasal, or up to like 37 subjects. But the common factor is, safe, without question. But generally, they all show cognitive and memory improvement. So that is the other thing. But we need to validate it with a large clinical trial. It's great to hear stories. We have a lot of stories of people improving. But I believe, really, to get credibility in the scientific community and with the regulatory people, you need to do a big controlled clinical study. It's very hard to do for Alzheimer's. The way how FDA has approved a drug, very expensive. You got to do PET scan and fancy things like that. But we are pursuing. So I'd say it's not enough to inform what's to be done. So I'm looking at how we can try to approach it from waveforms and brain networks. And gamma features really very prominently. So this is one presentation of the EEG. In a nutshell, you see the gamma. Now the left side is actually eyes open and right is eyes closed. The gamma is around 37 to 40 hertz. That's normal for young. And then as we age, the gamma slows down. And then when you have Alzheimer's disease, there's no peak. It's kind of, there's no set parameters. So your gamma is affected in Alzheimer's disease. Now, when you encode memory, and this is work coming out of UCSF with Dr. Muki's lab. He tried to look at the effect of gamma on epilepsy and memory encoding. When gamma, around 40 hertz, is present in the brain, the risk of epilepsy or seizure is lower. But in the meantime, the memory encoding goes up with the level of gamma in the brain. So gamma has this association with memory. And this is the famous landmark study done at MIT, which said that you flicker light at 40 times a second, 40 hertz, in a mice animal, you actually see a reduction of these markers, beta amyloid in the brain, the activation of this microglia to clear up this debris in the brain and so on. So it was really landmark. It woke up a lot of people. Although there was work already done, like I mentioned before, that's predated this one, but it really woke up people and what flickering at 40 hertz does with light. Then they added auditory and more response from the brain. They're doing a big clinical trial right now. Now the difference between what we do in photobiomodulation and this is like photic, photic, and you can say binaural as well, is that instead of stimulating the senses, sensory stimulation, what we do is going direct, penetrating through the scalp into the brain and influencing the waveforms and symptoms and so on. Okay, just to keep this short, there is, in, let's say, MS, multiple sclerosis, Alzheimer's disease, there's a buildup of glutamate, too much of it. There's a drug that tries to reduce it, like cytotoxicity. But gamma has been found to actually inhibit and reduce, this, because it activates GABA, which is an inhibitory neurotransmitter, to control this toxicity in the brain. Then this is an ongoing study that we are doing now at St. Michael's Hospital in Toronto. The left is MMSE is a way of measuring your cognition. There's a sham, there is which is control, and there is the active result. So the cognition goes up. But they are looking at the biomarkers through MR spectroscopy. So they use the scanner and see what the biomarkers are doing. And briefly found that the biomarkers that matter, like they use they observe a number of them. This is one of them. The marker that is associated with the declining brain actually reduced. They also did another kind of cognitive test called a thrill making B. Also, the time improved. Now, even in Alzheimer's, we can look at alpha. Now, this is very interesting. Those who are looking to brainwave patterns, fake oscillations, and so on, it's all about gamma, right? So much in the literature. But I got to tell you that it's not clear. And that is why in our future clinical study, I want to actually have the protocol include gamma and alpha. You have the first bunch of studies showing that, 40 hertz is best. But there's another study that came out of New York from a very respectable lab saying that, hey, we replicated MIT, we replicated your study on animals, it doesn't work. Then now there's a debate going on, there's a preprint, and say, MIT says it doesn't work because you're looking at different, comparing things differently. They are saying that 40 hertz actually work on the way it synthesizes with your, endogenous gamma. I don't know, it's going on, but the bottom line is not clear. Then a study that came out of China on animals too, very similar, mice. I thought this is a great study, but it doesn't get a lot of attention. They say 10 hertz is better than 40 hertz. No matter what you say, MIT, we did. And there's better, improved behavior in the animal model. There is more reduction in these markers, the amyloid beta. So it's up for debate right now. So to hedge the debate, I would say do both 10 and 40. I don't think we have time to show the study that we're doing now on the effect of pulse frequency on structures like the tubulin that polymerize into your microtubule that form the structure in your brain, your cells and your brain cells and all that. Is different. 10 hertz seems to, actually it's published, you can look it up, causes a transition in this, what's happening in the microtubules. And we are now investigating 40. I suspect 40 is consolidating it. So probably we need both. Watch for the literature. Now we position on a default mode network because it's a very important network. When you rest, you do nothing, no task involved, this network kicks in. And Alzheimer's disease, lesions and accumulation of the markers are mainly at the nodes of the default mode. This literature has been around for more than 10 years now. We put that because we believe from past work that location does focus a bit more on the nodes. So I would propose, putting more power in those particular areas. This is a study that we published in 2016, 2017, on 10 hertz. And MSSC improved. This was actually the first photobiomodulation study done on dementia. So there's merit to it, and that's before we started doing the 40 hertz. And here is a case where I did mention in traumatic brain injury that the photobiomodulation can regenerate the neurons in the brain. So the summary, 10 hertz, we may achieve similar, if not better, we don't know, reduction in the amyloid beta load, improved cognition, because it activates angiogenesis, which is creating new blood vessels, and neurogenesis, new brain cells. And now we're, in the neuroduo, we're putting into a clinical study. We'll alternate between 10 hertz and 40 hertz. This is a protocol once daily for people with those conditions. When you improve, reduce it so you don't get overstimulation. We like to have the vagus. We have a new device called a vagus nerve stimulation device, but it's actually difficult to do that in a controlled double-blind study in a clinical trial. But if you're not in a clinical trial, use it, because vagus nerve stimulation will also help. And there's some evidence in the literature too. So this is what it looks like. And there is, I think they're here actually, gamma core. It's handheld device. You can do this through electrical stimulation. We've got this being tested, and I say that with photobiomodulation, they pulse at, I think, 20 hertz, but we pulse at 100 hertz based on work done out of Harvard and Massachusetts General Hospital, and we found that 100 hertz frequency actually works better. But we'll see. You got to put through the clinical trials and stuff like that. But the device is available as a low-risk general wellness device, like our other devices. That is another way of looking at the vagus. I think why do we have one in the nose other than here? Because the vagus nerve actually starts in the brainstem. And the idea is there is evidence emerging actually from modeling work that we are doing with Baycrest Hospital, showing that actually the nose does activate all the way back to the hippocampus in the brainstem. So watch out for the literature. We just reviewed it last week. And then the other devices that we have, I just want to point out is we have pain devices. That's now registered with the FDA to treat focal pain. So we're not just all about the brain. And we have, want to touch on long COVID. Now, the long COVID, the main symptoms are fatigue, right? And I just get what the acronym stands for. But you stand up, you feel pain and all that, your heartbeat goes faster. And brain fog is the other big symptoms of long COVID. And our primary endpoint is going to be that. The study is being wrapped up right now in Florida. We're doing a pilot study to see how it goes. If it works well, we would go for the big pivotal trial and see, with the FDA. And so far, anecdotally, from other people, it seems to work, but we really want to do a big controlled study to be sure. And long COVID, I'll say, use the gamma at 40 hertz. It seems to help people with brain fog. It gets your brain to a higher functioning level, as I mentioned earlier, and on the default mode network, intranasal, and we have one. There's a big lymphatic junction at the back here, right, between the visual cortex and the cerebellum. So improving the lymphatic flow will probably improve it. And I believe it will be in my next paper. Lymphatic flow is actually one of the key factors in Alzheimer's disease. MIT just published about 2 months ago. So now I understand why flickering at 40 hertz seems to work. We didn't understand it before. Here's the answer. 40 hertz activates a neuropeptide that influences the lymphatic flow, called a VIP neuropeptide, and it opens up this astrocyte control barrier for better flow. So thank you, MIT. You also help us to explain why if I pulse it at 40 hertz, it's going to work too. So there's quite a lot of scientific joining stuff together and make sense of how to treat the brain. So that's the ongoing pilot study. I don't want to go through this, but generally that's being wrapped up right now in Florida. Then we did actually a study on acute COVID. This is not so much to do with the brain, just because we understand photobiomodulation, we understand what it does to the immune system, we understand what it does to microbiome, to for respiratory and so on. Because of the data we have, the paper is actually still being written up, but Health Canada has approved this particular device, the X Plus or RX Plus, to treat acute COVID. Now the thing is, you don't talk too much about it anymore because acute COVID is over, end of story. Now we're focusing on long COVID, but it has been approved for upper respiratory symptoms like flu, cold, COPD, and a bunch of other things. So it is a medical device. Now, the brain is complicated. We are exploring more. And I have to say that we really don't know anywhere near what we should know about the brain. It's the most complicated thing in the universe, right? And I say, okay, this is great. No, we're getting outcomes, but it really is just opening the gate to more research. And we want to personalize it by introducing artificial intelligence. And there is I think you can see we have a device called the NeuroPro that, especially if you're neurofeedback, you want to explore the influence of, different pulse frequencies on the brain. It gives you the ability to a phone to modify whatever you want to modify. You want to have beyond 40 hertz. This is being used for the long-term meditators to explore them going to an altered state. That is, data is being analyzed right now. It can, at a certain frequency, trigger for long-term meditators, not for me, for sure, but, for ordinary folks. But it can create this, they call it a bliss state. So it does have this effect on the brain, even healthy, almost like super kind of brain. So that is the kind of scope that photobiomodulation can cover. And okay, my time is up. Thank you very much for attending. Thank you.